Hi, I'm Matt Dunn.
And I'm Rachel Martini.
We're from MCDB 427 at the University of Michigan, and today we will be looking at figure 1511 from our textbook, which investigates the connection between polyadenylation and translation of mRNA. So first, let's take a quick overview of mRNA processing. We know that in eukaryotes, mRNAs undergo three main modifications. There is splicing, adding of the 5' cap, and polyadenylation, and we will take a look at each modification in turn.
The first modification we will review is 5' capping. Early in the transcription process, a cap is added to the 5' end of the growing mRNA strand. So this is our growing mRNA strand. The 7-methylguanosine is the cap that's added. This functions to protect the end from degradation, to enhance transport from the nucleus to the cytosol, to enhance translatability, and to increase the efficiency of splicing, which is the next modification we'll review. Remember that eukaryotic pre-mRNAs usually have introns and exons. To get rid of RNA that's not involved in expressing the desired protein, the extra RNA, called introns, which are shown here as the black line, are spliced out so that in the mature transcript you're left with just the exons.
Finally, mRNA transcripts are polyadenylated. So towards the end of the 3' UTR, there is a ribonucleotide sequence known as the polyA signal, and this calls for a downstream cleavage event of the mRNA at the polyA site. Then, at the site of cleavage, the enzyme polyA polymerase will come along and add a series of adenine nucleotides to the 3' end of the RNA transcript, and this is known as the polyA tail. Now, it's widely understood that these modifications are important for translating mRNA into protein. And if we look at the schematic of current theory, we can see that both the 5' cap and the 3' polyA tail bind to different proteins that form a complex with the 40S ribosome. However, this was not always apparent, and the experiment we look at today helped investigate the connection between this polyA tail and translation of the mRNA.
So before we get into the actual figure, we want to review polysomes and monosomes so we have a good understanding of them. To begin translation, a ribosome binds to the process mRNA. When the first ribosome binds complex, it's referred to as a monosome. You can see in pink here, you have the first ribosome binds to the mRNA, and this is a monosome. However, more than one ribosome can bind to the mRNA at a time. A transcript with many ribosomes reading it in tandem is referred to as a polysome. So in blue, you see a second initiation event, another ribosome starts translating the mRNA, and these are polysomes. The number of polysomes tells how well translation is occurring. An mRNA with many ribosomes bound to it is translating better than one with just one ribosome bound. To study the association of ribosomes to mRNA, you can create a polysome profile. To do this, you'd make a cell lysate, which will have monosomes, polysomes, and unbound mRNA, then use centrifugation to separate these out by size. To do this, you have a centrifugation tube that has a sucrose gradient. Here we have a low gradient at the top and high at the bottom. You add your cell lysate, and during centrifugation, the particles in the lysate will separate out by their relative densities, with lighter things going towards the top of the tube and heavier things towards the bottom. The relative mass in this case will be determined by the extent of the ribosome binding to the mRNA. mRNA that did not bind ribosomes, or free mRNA, will be towards the top, while mRNA bound to a single ribosome or multiple ribosomes will be towards the bottom. Then you would collect these fractions and assay for mRNA. In this case, we'll be looking at the absorbance at 254. So once you assay for it, you get a profile like this, where we get on the left, this is our low sucrose gradient, and that's our free mRNA, followed by the 40S peak, the 60S peak, and here is a monosome. On the left here, you see all the polysomes, so this is with 2-bound, 3-bound, etc. So that's what you'd see if you look at a normal polysome profile. However, our book does things a little bit differently. It flips it. So they always collect with the heavy things on the left side in the book. So that's as if they started from the bottom of the tube instead of from the top of the tube here. So we'll see the heavy things on the left are polysomes, followed by lighter things as you go towards the right, ending with the free mRNA.
So the data we're looking at today comes from an experiment done by Monroe and Jacobson. And the key question surrounding their work was, does polyadenylation of mRNA affect translation? So let's go through their method. The first thing they did was in vitro transcribe radiolabeled mRNA from a linearized plasmid template. And they synthesized mRNA both without a poly-A tail, and this was radiolabeled with 32 phosphorus, and denoted in our figure by poly-A minus. They also synthesized mRNA with a poly-A tail. This is denoted as poly-A plus, and it was given a radiolabel of tritium. Both variants of mRNA have a 5' cap, and both variants were added in equal concentration to a rabbit reticulocyte extract, which simply allows for translation machinery to act upon these mRNAs. Then, after they collect the extract, the authors perform ultracentrifugation and polysome profile, as we described earlier. The only difference is this time they're assaying for radioactivity, because keep in mind, each variant of mRNA has a unique radiolabel.
So part A of the figure is used to answer the question, are polyadenylated mRNAs translated better than non-polyadenylated mRNAs? To study this, they looked at the number of ribosomes bound to the mRNAs. mRNAs that are translated more have more initiation events, and are therefore more likely to have more than one ribosome translating them at a time. So they'll look like the blue polysomes here. That makes the number of ribosomes associated with the mRNA a good proxy for how well mRNA is being translated. This figure should look familiar based on the polysome profiles that we saw earlier. The difference here is that we're looking at radiolabeled mRNA instead of assaying for mRNA by the absorbance at 254. Remember that the sucrose gradient separates the affections out by size, so if we turn that on its side, you can see that the peaks we have in the graph correspond to the fractions we took off the tube. So we have these are our polysomes, followed by trisomes, disomes, monosomes, and this is just free mRNA. So here we're comparing the red and the blue lines, red being poly-A minus and blue is poly-A plus. The part we really want to focus in on is this polysome region, because that'll tell us the most about how much translation is going on. So poly-A plus has a greater percent of total mRNA that is polysomes than the poly-A minus, and that tells us that yes, polyadenylated mRNAs are translated better than non-polyadenylated mRNAs, and that the poly-A plus is associated with more polysomes than poly-A minus.
So the data over here in figure A can be a little bit tough to visualize. So now the authors are going to present the same information in a slightly different way with this plot over here in green. Now the x-axis is still showing our fractions from heaviest to lightest, but this time we're going to focus on fractions 11 through 28. Keep in mind that these fractions were the ones that corresponded to ribosomes in the form of polysomes. Now the y-axis is different this time. Now we're looking at the ratio of poly-A plus over poly-A minus mRNA. So this means that each data point greater than y equals 1, shown in red here, signifies that that fraction has more polyadenylated mRNA than non-polyadenylated. So we can see quite a bit more clearly now that the majority of these fractions contain mostly poly-A plus mRNA, and therefore it is helping with translation.
In part A, the researchers were using an mRNA that consistently had 68 nucleotide poly-A tail. They wanted to find out how long the poly-A tail needed to be to enhance translation. To investigate this, they created transcripts with varied poly-A tail length from 0 to 68 bases, and this figure shows the total amount of polysomes formed for the transcripts with varied lengths of poly-A tails compared to the one with the longest poly-A tail, which is 68 nucleotides. So, however much polysome was present here, they set that to be 100%. So that's this one here is 68 nucleotides is set to 100%. The rest are all percentages based off of that. So this figure shows that poly-A tail was not needed for translation. Here at the 0 adenosine nucleotides, we still get about 67% polysome formation, and there's no change between 0 and 5 nucleotides. However, we do get a large increase here from 5 to 30 nucleotides, and an overall trend that more association with ribosomes as poly-A tail increases in length. We don't know that 68 nucleotides is an optimal length, we just know that that's the longest one they tried.
So let's sum up what was shown in this data. In the free mRNA fractions, there was more poly-A minus mRNA than poly-A plus. And remember that free mRNA is not being translated. In the polysome fractions, meaning that the mRNA is being actively translated, there was more poly-A plus mRNA than poly-A minus. Furthermore, increasing the length of the poly-A tail correlated with an increased polysome formation. The authors also aimed to determine what the minimum length of the poly-A tail has to be in order to enhance translation, and they figured that it has to be at least greater than 5 nucleotides. However, the authors did not investigate a length of poly-A tail beyond 68 bases, so we cannot say for sure that this is the optimal length. They just normalized 68 bases to 100% formation. So the big conclusion from this data is that the polyadenylation of mRNA does enhance translatability, with the caveat that the poly-A tail is not strictly required because there was still quite a bit of polysome formation even without the poly-A tail. Thank you guys for watching, and go blue!